Good evening, everyone. My name is Dawn. Thank you for joining this webinar brought to you by ASQ Innovation Division. The topic of tonight's webinar is Innovative Ways of Controlling Microbiological Contamination and Recall Events in the Food Supply. This webinar will review current thinking in cGMP for food manufacturing and examine new considerations that will be necessary to maintain compliance. Methodologies for establishing and maintaining compliance with regulations while simultaneously enhancing business models and reducing operating costs will be discussed. The learning objectives are: understand what new software is available for operational efficiency, have an overview of new testing methods such as water content and rapid microbiological methods, understand what questions to ask in evaluating new testing methods, hear real-life case studies of new rapid micro-testing methods, both successes and those less than successful, and evaluate how a contamination issue could impact your business. This webinar will be recorded, and the recording will be available on ASQ Innovation Division YouTube channel. In addition, there will be 10-minute time after the presentation for questions and answers. Please post your questions on the Q&A panel of the WebEx tool. Tonight, our speaker is Mr. Kenneth Carnes. Mr. Carnes, ASQ CQA, possesses a master's in microbiology from Bowling Green State University in Bowling Green, Ohio. He has worked in the pharmaceutical, medical device, food, and dietary supplements industry since 1992. Prior to Regimus, he was working for B. Brown Medical initially as manager QC microbiology duplex, and then as site QA microbiologist overseeing all quality operations impacting aseptic production. Before moving to the West Coast, he held positions with Baxter in Maryland as microbiology manager, Novo Nordisk in North Carolina as QC microbiology manager, and Wyeth Pharmaceuticals in Pennsylvania as manager technical microbiology. Thank you very much, Kenneth, for accepting to be our speaker today. Without further ado, I would like to hand it over to you. Please take it away.
Okay. Thank you, Dawn. Appreciate that, and welcome, everybody. I appreciate your time. I know that it's not always easy to carve out time for something like this, but I really do sincerely appreciate each and every one of you being here with us tonight. So what I'm planning on doing right now is I'm going to launch into my talk, and one of my goals tonight is to make the quality people that are with us tonight very happy, because my overall goal is to make your life a lot more livable. And the way that we're going to do that is I'm going to show you some of the new or newer technical innovations. And I didn't say you're going to do less work, so don't take that. But the fact of the matter is that implementation of some of these projects can really make your life a lot more livable, a lot easier. And you'll see how as we go on. For any of the CEOs and plant general managers and or company owners, well, I'm going to pick on it a little bit tonight. So current regulations, if you go and you sit down like you're really having a hard time going to sleep, so you're going to sit down and you're going to read regulations, what you're going to find is the current regulations are actually really very nebulous. They don't give a lot of specifics. Now, they do that intentionally because many different businesses have multiple different business models. And so they want to be able to apply those regulations to the business model so that company owners, companies have the freedom in order to conduct their business the way they see fit, but at the same time be compliant with the regulations. The current regulations I've linked here, and the first question that I'm going to ask everybody is, you do have a quality unit at your company, right? Because you might actually be shocked at how many companies answer, no, we don't. Or they answer me, well, we've got a guy that's our director of manufacturing, and he's really a quality guy. And that's really nice. But you know, the regulations require that a quality unit exists at a company. That's actually written into the regulations. So for all of you quality folks out there, isn't it nice knowing that your job is actually federally mandated job security? I really have appreciated that throughout the course of my career. But the fact of the matter is that you need to have a quality unit. You must have somebody designated as your quality unit. And that goes for the very, very smallest of companies up to the very, very largest of companies. As an auditor or a compliance consultant, I've gone out and compared companies against the regulations as part of vendor audits, as part of audits for the USP Dietary Supplement Verification Program. And one of the things that I found is that there are surprisingly a huge number of companies that really don't have a designated quality unit. So this is the first thing I'm going to advise you. Please, if you don't have a quality unit, you need to get one. That's the first regulation that you got to follow. So the current practices that I've observed in the number of audits that I have conducted in the last couple of years, the first one is there is no testing, no quality whatsoever. So I call this the produce and pray method. And essentially, this is just, they bring their raw materials in, they composite them somehow, it comes out as a product, they bottle it, label it, and send it out the door. And again, you would be surprised at the number of companies that utilize this particular type of method. Now we contrast that to other companies that have strict incoming raw material testing, in-process testing, finished product testing, and all of these tests are conducted against quality-approved standards or specifications. And this I also call the FDA will never annoy me plan, because all of these companies that have these mechanisms in place, the FDA comes in, they look at everything, they say thank you very much, and they leave, which is one of our goals as quality people. The CGMP guidelines were never meant to be a government oversight program. A lot of people think that these regulations were put in place just to annoy and add expense and things like that. But really, they were designed to be a mechanism with which a product could be manufactured that would allow the FDA or any other auditing body to verify that the product was safe, pure, and effective. Now, we as quality people understand those three words. And we want to make sure that we can go to sleep at night. And the way that we do that is by doing our job and we verify that the product that we're shipping out to the American public is safe, pure, and effective. Now, another thing that I have noticed in my time as a consultant is that the overwhelmingly vast majority of companies that implement a good CGMP program or augment continuous improvement, anyone? Their CGMP programs, they initially hesitate at the financial cost. We got to hire some people? And they also kind of hesitate at the manpower hours that it takes to develop the programs. But ultimately, management sincerely appreciates it because what CGMP does is it provides a systematic methodology with which you can use to monitor processes within the company. And so what this does is not only does it give you control over the product, but for the people that are actually in charge and supervising and doing the overall mechanics process from beginning to end of the product life cycle, it gives you a very, very good control over what's happening throughout the course of your production process. Also, I just wanted to throw in there that for those food manufacturers, a good HACCP plan will identify the risk areas where microbes could enter a product. And those should be laid out as part of a good HACCP plan. So there are opportunities for improvement, and there are best compliance practices across food types. And so I'm going to talk a little bit about some of the software packages that I have seen throughout the course of my auditing. Now, I'm not going to go through every one, and I'm not going to go into huge detail. I'm going to go over two of the best ones that I have seen that I also find to be the most common. Okay, the first one is SysPro. Now, SysPro is a software that's designed for receiving, warehousing, and inventory management. And I have audited multiple companies that either have SysPro implemented or have it in the means or in the process of being implemented. Now, speaking as an auditor, SysPro makes my life easy. I can follow a incoming raw material. I can see where it was used, what lot, what batch it was put into. I can follow it all the way through to the finished product. And this is one of the mechanisms that an FDA investigator will utilize to investigate or to conduct an inspection on a company, is they're going to try and trace a raw material from beginning to end. In my opinion, and that's just what this is, one man's opinion, SysPro is one of the best for making this really, really easy audit. There are other ones out there. This is just one of them that I have seen that, in, again, my personal opinion, it really made my job as an auditor very, very easy. Another one that I've seen is Red Zone. And this is absolutely the most comprehensive manufacturing and in-process software that I've seen. You can take pictures of tablets or capsules and incorporate them right into the Red Zone software so that when you are in the process of manufacturing like a tablet or a capsule, you can put your in-process control data right into the Red Zone software. And you can follow, using Red Zone, exactly how fast the machine is running, how many tablets have been produced, how many pounds of compounding material have been produced. It really makes it very, very simple. And you can record all your in-process testing in there, like I said before, and you can record your finished product testing in Red Zone. And the thing that made it really, really nice for me as an auditor is when I sat down and they said, okay, everything's in Red Zone, they hand me an iPad, and I can actually just intuitively follow a product all the way through the manufacturing process. Made it a breeze for me to audit. And I have found in the course of my own day-to-day job that if you can do something like this with like an FDA investigator or an EU auditor or even just a client or vendor audit that's coming through your manufacturing area, if you can make it very, very easy, the audit is going to go very, very smoothly. And so the better you can make it, the more smooth you can make it, the better your audit is going to turn out. Okay, so now, one of the things I'm going to tell you is that all these software packages are dependent upon the amount of time and effort taken during the setup. So if you really want to make your life easy, don't rush through your setup. Okay, this is where the long-term part of the investment pays off. Don't rush the implementation. This is really where most companies fail to realize the benefits of software implementation. Because what they do is you got the CEO, the plant general manager, the company owner that's going to sit there and say, I've invested all this money in this software. Why aren't we utilizing it right now? And the fact of the matter is that you should plan tentatively to have about a year to get the software installed, to get the bugs worked out, to get people working with it, to get some super users trained so that they can train everybody else. And again, throughout this entire process, you're going to be working bugs out. You're going to do a validation of the software, right? You're going to have a protocol for each step of the validation, and you're going to have a final report that you're going to present before, that's going to be presented and signed by everybody involved in the validation process, including quality, and they're going to sign that protocol before you move to the next step of the validation. Now, this is, like I say, you're plan for a year. Don't be surprised if it takes 2, because for a software system such as like SysPro, it's a huge volume of data. Even if you have very few raw materials that you're using, it takes a long time to get all of those set up in the database and get everybody understanding the different steps they have to take. But once you take that time and you do all your upfront work, the long-term investment is going to pay dividends. Just because it's going to make it very easy for everybody to use. Everyone is going to be able to get information out of the system. The system is going to be valid so that when an auditor or an investigator comes in, we're going to very, very smoothly be able to present data to them, answer their questions, provide them with all the information that they need, and get them off your back. Okay, so there are opportunities and best compliance practices that I have observed through the course of my work here. Solids and powders. The one thing I can tell you with solids and powders, measure the water activity of your product. An AW of 0.95 or greater will provide sufficient moisture for the growth of bacteria and mold. So what this means is that there's moisture, enough moisture in your powder that microbes can grow. So an AW of less than 0.9 is going to prevent bacteria from growing, and less than 0.8 is going to inhibit the growth of most yeast and molds. Okay. There are three types of water activity measuring systems: resistive electric hygrometers, capacitance hygrometers, dew point hygrometers. The FDA recognizes all three systems. All three systems are completely acceptable to the FDA. However, again, each one of those systems must be validated to make sure that the data that they're generating is valid data. Okay, so you need to be able to show them your validation protocols. You write them, you approve them, you execute them, you write the report, you review and approve the report, and this equipment is going to be available for you. Okay, across the food types, we're going to look at some liquids now, and I'm going to tell you that in my experience with liquids, the biggest microbiological threat that I have seen to liquids is seams, joints, and welds, or even deep scratches. All of these things can harbor microorganisms. Now, you may think that, well, yeah, of course, but the fact of the matter is that when you are choosing equipment, not everyone looks at the seams and joints and welds and cleanability of a piece of equipment, because you need to be able to look at how is that equipment going to be cleaned, and it be torn apart, and it be easily put back together for the next batch that's going to be running on it. And I get into the very small seams, cracks. Are the welds such that they're not, what's the word I'm looking for, bumpy, that they're smooth enough that I can clean these things easily. Let me give you an example. There was a place where the plumbing had been installed in 1949, and it was a liquid delivery system. Well, what they did back in 49 is they actually ran the plumbing down into the floor, and then they poured concrete on top of it. Well, they were having terrible bioburden issues, and they couldn't figure out where the bioburden was coming from. And somebody finally got the idea in 2012, hey, let's run a camera down that pipe and see what it looks like. Well, as you can well imagine, after 70-some years in the ground, it looked like the lunar surface. And hence, there was a tremendous amount of bioburden that had taken hold and was growing up in that plumbing. And so they actually had to replumb the entire liquid delivery system. This did great things for their bioburden, but again, it took some time to get the upfront money approved. But in the long run, it paid dividends for them. And you got to keep in mind that there are certain microorganisms like Listeria monocytogenes. They can form really quick, and that bioburden is difficult to remove. And what they're finding is that pathogens such as Listeria monocytogenes have been showing increased resistance to a lot of different types of disinfectants. Now, I don't know of a Listeria yet that has been created that is resistant to high temperatures, or a product called MinCare, which is something that they push through water delivery systems in order to sanitize water delivery systems. MinCare is essentially an acetic acid plus hydrogen peroxide. And there's no bacteria yet that is resistant to those materials. But a lot of people will use just like a dilute chlorine bleach or something like that. That's bleach is great, yes, but it also corrodes a lot of stainless steel piping. So unless you have glass piping, which I have also seen, bleach is not a really good option. There's a lot of organisms, and one that comes to my mind is Ralstonia piquetii. Ralstonia is not known to be pathogenic, but the biofilm produced by this organism is extremely difficult to remove. It is just like chunking concrete. It is very, very stiff. And then there are also portions of it that have a gossamer veil kind of appearance, almost like a jellyfish. And between those two types of biofilm that it produces, it's a mess to try and get out of your plumbing. So again, prevention is the best way to control this, and that is to be able to get access to all the different liquid delivery systems that you have to clean. Testing techniques and rapid response. Okay, now we're getting into the manufacturing process. And what is rapid response? And rapid response would be something like you determine, oh, there's an OOS, there's an out of specification test result. Now this could be a incoming raw material test, this could be an in-process test, or this could be a finished product test in the laboratory. These things require a rapid response, so do deviations. And in the very worst case scenarios, you have CAPAs. And each one of these different things requires a very, very thorough investigation. So this is kind of a worst case scenario. Now you have a product, you have an in-process product, or you have a finished product that has exceeded tolerances or gone beyond the specification. And so now you need to do an investigation. So now, quality people, plan out your investigation before you even start. You should have a good, thorough SOP, because each one of your investigations should by itself be a standalone document. What do I mean by that? You're going to put in all associated emails, correspondence, and meeting minutes, okay, into a folder or someplace that is a centralized location. This could be an electronic folder. It can be actually a folder in a file system. I don't care, however you want to put it together. And, oh, did he say meeting minutes? Yes, he said meeting minutes. Because even if you have an out-of-specification test result and you are requesting somebody in quality to initiate an investigation on that out-of-specification test result, yes, it's in procedure that it has to be done, but you should also document when it was initiated. If you're doing a very, very large CAPA investigation, you've had a major problem, and now you're doing a very large CAPA investigation, and you've got your CAPA team together, and they're going to come together, and everyone's going to sit down, and you're going to do some brainstorming, and you're going to use some of your 6S tools in order to solve the issue. You want to have meeting minutes to document exactly what happened, who was there, what was discussed, what were the results of the discussion, who got assigned what tasks in order to conduct the remainder of the investigation. Any emails, you're going to put all of these things into a centralized folder to capture everything for the investigation. Purchase records. So for instance, if you have to replace a pump, you're going to want to put in the purchase records to show that, yes, indeed, you did in fact buy the pump that was called for as the corrective action. So you want to put your purchase records into your investigation folder. You want to have copies of the qualification or validation reports. Now, a lot of people say, when is a qualification or a requalification and validation necessary? Well, for those of you that have been in quality for a while, you're going to know that if it's a like-for-like change, so if you have a pump went bad, you buy the exact same pump, Do you need to do a requalification? No. But if you want to do like a mini requal, I have seen those done, and those are very, very effective at showing that, yes, the corrective action was completed and the machine still functions within its validated parameters. On the other hand, pump goes bad, that pump doesn't exist anymore, now we got to get a new pump. Does that require qualification? Yes, it does. And do you have to go through the full IQ, OQ, PQ? Best practice says yes. Just because you have essentially now exceeded the original parameters of the equipment's qualification, and you've changed something that's in the qualified or validated piece of equipment. So we need to move from there and do a requalification. Continuing on with the components, we need to have training records. We need to have copies of current procedures. Why is this important? Well, because if the investigation takes place in 2020 or 2020, and the audit doesn't take place until 2024, your SOPs may have changed 4 or 5 times. And so it's always a good practice to have copies of the procedures that were current at the time be included in your investigation folder. And that way, the auditor or the investigator can very much see, okay, they were in fact complying with their procedures at the time, because this was the active procedure. So that's a really important thing. Any protocols or protocol reports should be in your investigation folder. So these should be copies, not the originals. But if you have to requalify a piece of equipment at the end of your investigation, all of that should be in the protocol. All of that should be captured in your investigation folder. You should have copies of the protocols and the final reports signed off by QA. If an investigation is ongoing at the time of an audit, it's really very critical to have information to present to an auditor that shows exactly what has been done up to that point and what the investigative plan is to close out the investigation. And the reason for that is I have been caught myself in the middle of an investigation, and the auditor asked me, so what have you done? I was able to present him with the initial meeting minutes, a copy of the open investigation form, and the procedures that were active at the time that everything took place, and training records to at least show that the individuals who were conducting the investigation and who were involved in the activity at the time of the auto specification were in fact trained and ready to do that work. And then I was able to show them the plan. Here's what's going to happen from here on out. Here's how we intend to conduct this investigation. The auditor at the time was very pleased to see that. So I've also been in situations, being the auditor, where I've asked, so what's the investigation? What have you done so far? Well, we just started, and so everything is still ongoing. Okay, great. What have you done to this point? Yeah, and they'll be able to tell me verbally, but they won't be able to actually present me with any information. So I really don't have any idea as an auditor how their investigation is going. And so you want to be able to make that auditor, whoever it is, you want to be able to put them at ease and show them that your investigation is being handled and being handled thoroughly. Okay, now, quality people, I'm going to talk to you for a second. Don't predetermine the disposition of the investigation. If you're with an auditor, and especially if you're with an investigator, don't tell them something along the lines of, well, I'm sure it's going to be fine, or it's really no big deal. It's always the investigation is ongoing. Don't have it also predetermined in your mind that, oh, we're going to find that this is nothing, and so the product is going to be fine and be able to go out the door. Because it has happened where I've gone through 80% of an investigation, everything was fine, and I was thinking, yeah, this is going to be able to go out the door. And then some other critical piece of information comes to light, and I have to rescind that. And if I've already told my boss, yep, this is going out the door, Well, that becomes a very uncomfortable conversation, and nobody likes that. So don't predetermine the outcome of the investigation until the investigation is actually completed and you've signed off on that disposition. Another thing I'm going to say is do not be swayed to decide how the disposition is going, or how the disposition is going to go. And this is where I get to pick on some of the CEOs and industry owners, is because they're going to sit there and you are going to hear them say, I need to get this out the door. It's very, very important that we get this out the door. We need to sell this product. You as the quality person, if you haven't been yet, you will be, because there's two types of people in quality, those that have experienced this and those that will experience this problem. And this is when the production folks put pressure on you to disposition a product to send it out the door. It's going to be fine. Hey, yeah, it's LOS, but you know what? It's not that LOS. We can still send it out the door, right? Come on. No. This is where you have to be the bigger boy. And if the disposition of the investigation and the data and evidence tells you that the investigation is not one that you can release the product, you don't release the product. Because our job as quality people is to provide the American public with products that are safe, pure, and effective. We've got to keep that in the back of our mind. And we can't let that bottom line get in the way and influence our decision as to how we're going to conduct our investigations and the disposition of our investigations at the end. Okay, now, in order to help you with your investigations or prevention of potential investigations, there are some rapid micro methods that are out there that have been accepted by the Food and Drug Administration. So I know one company that actually they tested their water just before it was added to the product. And they did not actually put the water into the vessel until the results came back from the micro lab saying, thumbs up, water is good, go. And I found this to be a very, very good way of doing it. So I was like, hey, how are you doing this? Well, they had a rapid micro method that they had validated, and validated well, where they could actually get results back in about 4 hours from the time that they took the result to the time, took the sample to the time that they had a result that they could get back to production. With some planning with the production folks, they managed to get that working like a well-oiled machine. Hey, we're going to be adding in about 4 hours. Here's your sample. Thank you very much. Here's your result. Production, go. And so there's a lot of advantages to these rapid micro methods. Faster release of raw materials. A lot of people function in a just-in-time manufacturing process, and so they want to get get their materials in and get them used as quickly as possible because inventory is overhead, right? Well, you want to get those raw materials in, you want to get them tested, you want to get them tested quickly, and you want to get them being made into product. So then also faster transfer of in-process work from one stage of production to the next. That goes to my example of testing the water before you move to the next production process. And this is one of those places where red zone really shines. A production person will be looking at their iPad, and they're going to see that this test has to be completed before the next stage in the production can be taken. And so they actually stop, because that's what they're trained to do. They actually stop and say, hey, I don't have this result in here. And they're on the phone with the quality control people going, hey, I need that result so I can continue on. Oh, here you go. And the quality control person in the QC lab enters the result from the test, and then the production can continue. And so this is a really, really good way to make sure, again, that during the course of the manufacturing process, nothing is happening during the manufacturing process that could potentially adulterate the product. Again, this is going to reduce your inventory requirements. And what this means is a lot of products have to have some kind of release testing in order to get them out the door and say, okay, quality, Quality has now approved this product. We can actually put it on the truck and ship it out. And you want to minimize that time that it's sitting in your warehouse as much as possible. And that time that it's sitting in the warehouse, of course, is money. And so you don't want to spend money just having products sitting in the warehouse, of course. And so you want to be able to get it out the door as quickly as possible. Now, another thing that a lot of people don't even take into account is the ability to recover quickly from a contamination event. So if the sample gets taken just before it gets added to the product, Micro takes it back, tests it, says, oh, no, we got to run a cleaning cycle before we can use this water because it's OOS. You've just saved yourself a ton of money because you got raw materials sitting there, they're waiting to be made into product. But had it been made into product with contaminated water, then you'd have had raw materials that you're throwing away. And in this case, now you've saved those raw materials, you run your cleaning process, redo the test is good, now you go on and you've got quality product that you can show has been released and is ready for use. And so the ability to just recover from a potential contamination event, or if you have a raw material incoming that's heavily contaminated, like a botanical, and you don't want that botanical to be put into a liquid where the liquid is going to propagate microorganisms, and so you want to be able to recover quickly. So you want to be able to test that, get that test information back so that you can continue on with your manufacturing process as quickly as possible. Have the rapid micro methods been accepted by the FDA? Yes, they have. One of the most strict tests that the FDA has, and I'll jump into the pharmaceutical industry for just a minute, is sterility testing. This is a very, very critical test just because when you have a sterile product, that implies that the product is going to be injected into a patient directly, which bypasses the greatest defense mechanism that the body has, and that's the skin. And so there are a whole lot of considerations that you want to take into account before you decide upon a specific rapid micro method. Okay, what is the volume of the test material? Are you testing 1 milliliter? Are you testing 50 milliliters? Or are you testing a full-on liter? What's the system capable of testing? Why is this important? I'll talk to you about that here in just a minute. And what are the training requirements of the system? You know, does the person using the system just need high school education? Or is this something that a PhD microbiologist needs to operate? And there are systems that encompass both realms. And what's the cost per test? That's something that you got to take into consideration. And when you're doing this, be sure that you evaluate the reduced time in inventory, because a sterility test takes 14 days. So you finish making a product, and it's got to sit in your warehouse for 14 days before it can be released, because you got to get your sterility results. And so you want to be able to reduce that time in inventory. So in the example that I'm going to use, there was a company that had to do sterility in order to release their product, and sterility, it's 14 days by the what I call the traditional method. And so the rapid sterility method that was being investigated required a 4-day incubation. So you can imagine how happy the CFO was when we told him, hey, we can reduce the time in the warehouse by 10 days. That's a huge cost savings, especially considering the volume of the product that this company was making. And so he was really excited. He thought this was going to be great. Okay, so the volume that one test setup using this new method was able to test was about 50 milliliters. And so when we did our first initial evaluations of the test method, it didn't occur to us that the material that we were going to be testing was actually a full liter. So when we got to a point where we had to evaluate, was this a cost-effective method for testing, we had to take into consideration that we had to take 1 liter divided into 50 ml aliquots, and then each one of those separate 50 ml aliquots was a separate individual test. And so the cost per test became much more than the traditional method. And it even exceeded the savings that we would have seen by reducing the time and inventory. And so ultimately, even though the test method itself was viable, worked great, it detected 1 CFU, it was a wonderful test method, we had to throw it away just because you can't test a 1 liter product 50 mils at a time. So when you take, if you really want to scare yourself, here's a fun little exercise for you to take. Look at your production costs. Look at the overall production costs. Break it down to production cost per process or per process step. Look at what if we had microbial contamination in this step, what all would it contaminate, and how would that contaminate everything down the chain, down the manufacturing line. So you want to evaluate that cost at each of the production steps. So an example would be you have an incoming raw material. If you have an incoming raw material, what's going to happen? The product is going to end up contaminated, all your equipment is going to end up contaminated. It's a mess. And so you want to know that as quickly as possible before you even use the incoming raw material. A lot of people have asked me, hey, is it okay if we start using a product while it's on test and then release it once the test results come in? Well, I would ask you to evaluate how far down the production process is it going to be before the results come in. Because if you have a contaminated raw material and the product is absolutely complete by the end of the test, when you find out the raw material is contaminated, you've got a lot of product you're going to be throwing out in the trash. So ultimately, I find the best policy to be no use of anything until the QC results are in and into either into a data sheet, your batch record, or something like Red Zone. If a raw material gets contaminated during weighing, what's your cost there? Or if it gets contaminated as it's going into the blender. And look at quality people. You should be looking at what preventative steps are you taking to make sure that something doesn't get contaminated during weighing. Are you weighing in a way, in a booth, in a sample booth? Well, I would hope so. But I have seen people do their sampling and weighing just out in the middle of a warehouse. And I do not consider that to be best practice. So all of these have to be taken into account. And the reason that I mention this is because there's a study done in 2015 by Ohio State University that evaluated the cost of microbial contamination in the food supply. Now, this study took into account things like the cost of medical treatment, lost productivity, illness related to mortality, and overall, taking into consideration all these above factors, $55.5 billion? That's like government level money. And that is a significant chunk of change. And so you really, as quality people and as business owners, need to evaluate your costs per step and look at the controls that you have in place. How quickly can you get information? One of the things that I'll tell the quality people is look at it from the back end. Look at it from the end of, okay, now I have a problem. How do I get information that is going to help me determine whether the problem is catastrophic and we have to throw the product away, or what information can I obtain that is going to help me prove, beyond a shadow of a doubt, that the product is still adequate for use. And so you want to think about it from that back end. What information are you going to need? And then you can start looking at working your way backwards, and then look at the systems that are available to provide you with that information. How quickly can that system provide you with the information? And then how quickly can you put your report together and come up with your disposition? That's usually pretty quick. Okay. Last thing that I just want to mention really quick here in this topic is how the recent coronavirus pandemic impact. And right now, I've seen some pretty staggering numbers. And the reason for this mainly is because I have several clients that at the beginning of the pandemic, they said to us that, no, we're considered essential personnel because we're making a dietary supplement. No, we're considered essential personnel because we're making an over-the-counter pharmaceutical. But in almost every case, each one of those people have had to say, well, we're shutting down because we have so many people sick that we just have not enough personnel in order to carry us through. And so the problem really for those companies is not so much the impact from the supply and production management process, but from the personnel aspect. And so it will be very interesting to see how that all comes out. Now, we're going to pretend for a minute that the worst case has happened. You've determined that there is a need for a recall. How do you make the recall smooth and efficient? And so you're going to put together your recall plan. All of you have a recall SOP, right? Yes, shake your heads yes, because you do. A recall plan is absolutely critical because when recalls happen, having been there, you will panic. And so you want to be able to think as clearly and as straightforward as you possibly can. So the first thing that you need to do is affirm that your recall plan is treated as a preventive control. And in your procedure, you're going to describe when a recall plan is required. What has to happen in order for a recall plan to be initiated? If you put those in the procedure, it's going to make it a whole lot easier for you. You're going to list the procedures that are applicable to the recall plan. So that's going to be like customer service, communication with the FDA. You do have a procedure for communicating with the agency, right? Disposition and rework procedures, investigation procedures. Remember that investigation that we talked about? Yeah, we need that. Warehouse and inventory procedures. All right, and then we're also going to list out our food defense plan requirements, and that's going to include and nobody likes to think about this, but the identification of areas where the manufacturing process is vulnerable to intentional adulteration. Did somebody do this on purpose? And that's an important thing to consider. Some common recall practices. Now, in your procedure, there should be defined roles and responsibilities for the recall. Everyone should know exactly who's going to be on the recall plan committee and what their job is in performing the recall. Of course, we talked about procedures to determine if a recall is needed. You also should have somewhere in there a list of contacts for external notification. So, for instance, if you send all of your products to Amazon, you need a process that somebody can use. Who do you get in touch with at Amazon? Who do you tell? What information do you provide them? Et cetera, et cetera. Do you actually talk to customers? Do you call individual people? Excuse me, Mr. Smith, but the product that you bought from us, we have to recall it. Can you please send it back to us? How are you going to do that? You should have procedures and training for personnel that are going to be involved in that. You should definitely have lot numbers, identifications, and descriptions of the product that's going to be involved in the recall, and effectiveness checks. Now, what is an effectiveness check? Everyone remember plan, do, check, act? Yeah, the domain. Yeah, that's what that is. So this is an effectiveness check just to make sure that everything is going to be going, that once the recall is in place, you know how successful your recall is being. And you should have standard forms that you're going to use to record your data from the recall. And one of the things that I have found very, very useful is to have information notice templates. So you may have preset wording that your customer service representatives are going to use. You may have preset wording as to exactly what your email to the FDA is going to look like in the event. Okay? So these are some tips and pointers that are going to help make that whole process easier for you. I hope none of you ever have to experience that. It's not pleasant. It's very stressful. It's one of the worst things that can potentially happen. So I hope that none of you ever have to have a recall. And I hope that you're all thinking about how are you going to implement effective processes and procedures that are going to prevent contamination and make it so you never have to have a recall. Thank you all very much.
Thank you very much again, Mr. Carnes, for your presentation. Please post your questions in the Q&A panel of the WebEx tool. I shall remind everyone that you can find the recording of this webinar on ASQ Innovation Division YouTube channel. If you have any questions, you can post your questions in the Q&A panel of the WebEx tool. I have one question. So actually, it's from me. So you mentioned that OOS and kappa and deviation, they are the testing responses. Or can you elaborate on them?
Yeah. I'll tell you that what the agency, what the FDA expects when you have an OOS, or you have a deviation, or even something very major goes wrong and you have a capital assignment, the agency expects you to react to those things pretty fast. And so each one of your procedures should tell you what time frame each one of those, an OOS has to be responded to in X number of days or X number of hours. Some people say 24 hours. It's got to be identified in the investigation open. Some procedures don't, but you should have somewhere in your procedure how quickly you're going to react. And the faster you can get the investigation open, the faster you can get data, the faster the investigation can be closed, the more efficient your process is going to operate overall. So you want to just be able to have that really, and that was all part of the rapid micro method testing. And so it's one of those situations where you can get that data back that shows that, no, there was no contamination, or no, it was a false positive, or something like that, or no, we definitely have an issue. The more efficiently your investigative process is done.
Thank you very much.
Yeah, because I'll give you one other little real life example. FDA came and was doing an investigation at one place that I was working, and they found that the kappa had been open for 8 years. They did not consider that to be a rapid turn.
Thank you. There is one question that just came in. So what about using ATP rapid test in food or pharma as a very rapid tool?
ATP testing has been accepted by the agency. The only thing that I would caution you in regards to ATP testing is be sure, first of all, that your ATP test is testing something that you are actually, that could actually potentially be a contaminant in your area. So the reason that I say that is somebody that was basically making a vitamin C tablet was testing for residual protein. That didn't make sense to me. But it has been accepted by the agency. Now, another thing that you got to keep in mind is that each one of the manufacturers of ATP test kits have their own system and their own measurements and values of here's what the ATP kit is going to show. And so this is considered acceptable and this is considered not. And so I had to get used to, as an auditor, going to one place and seeing a value that might be 12. Okay, 12 is acceptable. I go to another place and it's 140. I'm like, whoa, wait a minute. This test kit, 140 is acceptable. So just keep that in mind. And again, I would definitely do a qualification of the testing and put together a protocol and write a report before you actually rely on the data from that ATP.
Thank you. If there isn't any more questions, so we can conclude this session.
Well, again, I'd like to thank everybody for taking the time to be with us tonight. And have a great evening.
Thank you very much, Kenneth, for your time and presentation too. And thank you all for joining this webinar. Have a very good evening.